This is critical thinking in the cell transport lab, and while you're going to be doing activities using the PhysioX CD, here I'm not going to discuss the CD but kind of look at some practical applications. So let's look at this first question and take a read this question. Here is the second one, which is pretty similar. Then we have this question, which is again on transport mechanisms, and here is another one. So let me go back to the first one where it tells you you have a handful of hard dried beans which you want to cook, and before you cook you want to make them swell and soften them up. So what would you do? Would you put them in a bowl of distilled water? Distilled water is pure water which has no solutes at all. Or would you put it in a bowl of concentrated salt solution? In other words, there's water with a lot of salt in it. Or would you just leave them overnight without any fluid? So obviously this choice, you would not choose this one. So let's look at these two. Now remember, when you have two, let's say you have two solutions. So let's take the first one, which is this, where you have a water with no solutes at all. So this is 100% water. So this is 100% water, and so we call this, which has no solutes at all, we will say this is hypotonic. And here let's look at the second one, which is concentrated salt solution, and let's say we take the same volume of the solution up here, but it's concentrated. So let's say up here this is all salt. So we have, so this was 100%. So suppose this was 100 ml, all of it is water, so 100% water. Here we have 100 ml of the fluid, but let's say we have only 50 ml of water and 50 grams of salt. So if you look at it, can you see the water concentration here, because it's half, is only 50%. So because of the presence of solutes, you can understand that such a solution which has more solutes than this one will be called hypertonic. Okay, now water always moves from a hypotonic to a hypertonic area. So if we were, suppose we put these two solutions in a test tube, and let's say we take a test tube like this, and it is separated by a semi-permeable membrane. On one side we put the hypotonic solution, and on the other, that means distilled water, on the other side we put the hypertonic solution, which was the salt solution. Because osmosis is the movement of water, what will happen is from here water will move to this side till the concentration of water is equal on both sides. So let's say it comes to 75% water on this side, 75% on this side. So obviously the volume on this side will increase. Now let's see how we'll apply it to this. So here we have hard dried beans, right? So they don't have any water in them, but they have a lot of solutes inside. So which is the best way to make them swell? The best way would be if you put it in a bowl of distilled water. So you have this 100% water over here, and if you put this inside here, what will happen? Water will move from the bowl into this dried bean. So it will go inside and the bean will swell. So therefore, in order to make them swell, this is what you will do. You'll put them in a bowl of distilled water. You can see it's going from a hypotonic to a hypertonic area. See, the bean is hypertonic because it has all solutes, very little water in it. If you put it in a concentrated salt solution, well, some water will go in, but after some time not much will go in, because even this bean has a lot of solutes in it. So therefore you can see that the correct answer to this question would be you would put it in a bowl of distilled water. Now just to give you another practical application, let's look at this question. When a person is dehydrated, you set up an intravenous drip to replenish the fluid and electrolyte loss. So here what I would like you to see is that it tells you that, when you vomit or you have diarrhea, you are losing both fluids and electrolytes. So here's something that you should know, that Normally your body does lose electrolytes. It's normal to lose some amount of electrolytes. Electrolytes are like sodium, potassium, all of those, chloride, bicarbonate. When you pass urine, you lose some electrolytes. When you sweat, you lose electrolytes. If you vomit, you lose electrolytes. If you have diarrhea, you lose electrolytes. Now if you lose a lot and you lose a lot of fluid with it, you obviously have to replenish it. And it asks you, what would you give this person, a hypertonic, an isotonic, or a hypotonic? Isotonic means it is the concentration is the same as that of blood. Hypertonic means it is hypertonic as compared to blood. Hypotonic is it is hypotonic as compared to blood. So the correct answer is this, and let's see why. So let's say this is a blood vessel, and inside this you have cells, and I'm just going to take some red blood cells. Let's say you put a hypertonic fluid inside. So that means the fluid has less water, more salts. So you push that fluid in over here. So now that fluid has come here. So this is hypertonic as compared to the red blood cells. So what will hypertonic solutions do? They tend to draw water towards themselves. So water will come out of the red blood cells out here, and the red blood cells will begin to shrink. So they will crenate like this. So you don't want that happening. Therefore, you will not give hypertonic fluid. Next is hypotonic fluid. So would you give hypotonic fluid? So again, taking the same blood vessel with the red blood cells inside. So now you give hypotonic fluid inside here. So remember, what does fluid do? It goes from a hypotonic to a comparatively hypertonic area. So as far as the red blood cell is concerned, the fluid outside is hypotonic. So from here, the fluid will start going into the red blood cell. So what will happen to the red blood cells? They'll get larger and larger. They will swell and they'll finally rupture. So therefore, you don't want this either. Now what happens when you give isotonic fluid? So here and here are the red cells inside. The fluid has the same electrolyte or solute concentration as that of the cells. So fluid will be given here. There'll be no movement of fluid into or out of the cell. So they'll remain normal, and you'll just replenish the fluids and electrolytes in the plasma. So that's really important to know. Now based on this, you can see, and when you do your experiments, your activities, you will see that you're asked a question on the relationship between solutes and osmotic pressure. Now you can see fluid moves from a hypotonic to a hypertonic area, and so we know this. But what if the fluid was not allowed to move? So let's say we have this huge sort of beaker with a semi-permeable membrane, but the volume is constant, and this side you have more solutes, and this side you have less solutes. So this is hypo and this is hyper. So fluid will tend to move from hypo to hyper. Now if the volume is not allowed to change, what happens? The pressure inside builds up, and that pressure is known as osmotic pressure, because that's what draws water towards it. So you can see wherever there are more solutes, the osmotic pressure is much higher, because that means it's wanting to draw fluid in. Wherever the solutes are less, the osmotic pressure is lower. So that's the relationship you can see between solutes and osmotic pressure. So that's something you need to learn from the activities you do. Let's look at this question. This is a fairly simple question. This is showing you phagocytosis. You can see this large sort of a molecule or large substance which has been cell debris, which is taken inside the cell, goes in, forms a vesicle, and then this is engulfed by lysosomes and they destroy it. So it asks you, first you need to identify what is it. The correct term for this is phagocytosis, which is a type of endocytosis, but it's best to write the exact type because there are other types of endocytosis. So this is phagocytosis, and it asks you which cells are capable of the process. So the cells which are capable are both neutrophils and monocytes. So both are in this answer. So this is the correct answer. Never just red blood cells are not capable of doing that. Don't just write white blood cells, because not all white blood cells are capable. It's only certain white blood cells, and those are neutrophils and monocytes. So this is the correct answer for this. It's very important to look at questions like this. And lastly, this question is on active transport, and it asks you what would be the result of transport of sodium and potassium across a muscle cell membrane if the sodium potassium pumps are inhibited. Remember, in order for transport to occur in sodium and potassium, not only do you need ATP, you also need the sodium potassium pumps to be plentiful and to be working properly. So therefore, if even if you have enough ATP but the sodium potassium pumps are not working properly, then transport will decrease. Or if sodium potassium pumps, the pump number of pumps is decreased, then this will also decrease. So both have to be available in order for transport to occur properly and in the right proportions. Okay, so read your questions very carefully and then choose the right answer.